Hello guys, welcome to NNL Learning TV. So today we are going to learn about the 64 possible codons and each amino acid allocated to it, and we are going to learn some tricks and shortcuts to go about it and get it in no time. So let's go into it. First of all, let's look at the genetic code and codon. So the genetic code is a sequence of nitrogenous base in the DNA that showed or indicate the type of amino acid that the cell makes. It is a known fact that the DNA carries the genetic information, and this information is based on the nitrogenous bases in the DNA molecules, which makes it possible for us to determine its amino acid. So let's look at the codon. Here are the sequence of 3 nitrogen bases that specify the different amino acids in the DNA molecule. For example, we have the UAG and the AUG. For start codon, there are 64 possible codons and 20 amino acids, for which we are going to learn going forward in the table, and we'll be getting to find some simple tricks to get it done. So let's look at the start and stop codons. For the start codon, we have the AUG, which goes for methionine, and over there at the start codon, the ribosome binds to the messenger RNA at the translation level of the protein synthesis. So now let's look at the stop codon. The stop codon, we have UUU, AG, AGA, that's the trick for it. And at the stop codon, this is where the release factor binds to the stop codon, which helps in terminating the translation and helps the release of the complete polypeptide from the ribosome, and that makes the translation to end. So let's get to the table where we have our codons and respective amino acids. So we have the rows and columns over here, where we have about 16 boxes, of which each contains 4 codons. Okay, so on the left side of the boxes, we'll spread out the USAG, that is UCAG, and we'll spread it in each of the boxes. The U will take 1, C will take 1, A will take 1, and G will take 1. At the top, so we'll do the same. USAG, U first, C, we have A, and then the G. And at the right side of each boxes, we'll be putting USAG in each side of the boxes, of which will be the third part of the codon. So the left side is the first part, the top is the second part, and the right side is the third part of the codon. So we have UCAG on each side of the boxes at the right side of the table. So let's get to it. So we know that the codon has 3 parts. The left side of the boxes is the first part, the second part is the top, and the third part is the right side. So starting with the boxes at the left top corner, you'll be putting U there, and then the U at the top will come, and then the U at the right side will be coming. So the second U and the third U, and you will come down and put the first U, the second U, and C. So you see how we go about it. The first one is from the left part, and the second one is from the top, and the third one is from the right. So we're putting it inside the boxes, arranging them in an orderly manner. That's how we go about it, getting to know each of the codons, so you don't get to memorize each codon in your head. So we'll go to the fourth row of the second column. We have G to be the first part, C to be the second part, and U to be the third side from the right side. We come down and get G at the first part, C at the second side, and C again at the third side. For the third one, we have G, C, and A respectively, like how we did the first ones, and then after. So we continue to fill all of the boxes. So let's go to the fourth row of the fourth column. We have G, U. The second one we have G, C. Then we have G, and A from the right side. So we'll be continuing till all the boxes are filled. So let's start with the prospective amino acids. So the first one we have is phenylalanine, which takes care of the first two codon of the first bus. In the same bus, we have leucine, which takes the two other down, and then leucine takes the whole bus for the second row of the first column. Then on the third row of the first column, the first three will take isoleucine. The last one is methionine, and that methionine is the start codon, makes the ribosome to bind with the messenger RNA. Then the last bus of the first column, we have valine, which takes the whole bus. So now let's fill it all out and get to where the trick is, and we'll be learning them column by column how to go about it, the tricks we will use, and some fast way to go about it. Trust me, you will get to memorize 20 amino acids with 64 possible codons. So let's get on it and start with the first column. In the first column, we have phenylalanine, leucine, isoleucine, and methionine. So the trick for it is Phoebe, let Isaac meet Valerie. Phoebe, let Isaac meet Valerie. So Phoebe is for phenylalanine, let is for leucine, Isaac is for isoleucine, mate is for methionine, Valerie is for valine. Phoebe, let Isaac meet Valerie. Phoebe, let Isaac meet Valerie. Let's get to the second column. In the second column, we have serine, proline, threonine, and alanine. So the trick for this one is serine, promise, theophilus, alanine. Serine, promise, theophilus, alanine. The serine is for serine, promise is for proline, theophilus is for threonine, alanine is for alanine. So serine, promise, theophilus, alanine. So trust me, it's very, very easy. Now let's get to the third column of the boxes, which is the column of A. You have tyrosine, stop, histidine, glutamine, asparagine, lysine, aspartic acid, and glutamic acid. Don't mix the glutamine with the glutamic acid. So the trick for this one is tyrosine, stop, his glue as lysine. Aspired glutamic acid. Tyrosine, stop, his glue as lysine. Aspired glutamic acid. Tyrosine is for tyrosine, stop for stop. His is for histidine, glue is for glutamine, as is for asparagine, lysine is for lysine, aspired is for aspartic acid, and glutamic acid is for glutamic acid. Tyrosine stop his glue as lysine aspired glutamic acid. Tyrosine stop his glue as lysine aspired glutamic acid. So in this last column, we have cysteine, stop, tryptophan, arginine, serine, arginine again, and glycine. The trick for this one, we have cysteine, stop, trying, arbitrating, serine, and glycine. Cysteine, stop, trying, arbitrating, serine, and glycine. Cysteine, stop, trying, arbitrating, serine, and glycine. You have the cysteine for cysteine, stop for stop, trying is for tryptophan, arbitrating is for arginine, serine is for serine, and is for arginine. Glycine is for glycine. Cysteine, stop, trying, arbitrating, serine, and glycine. Thank you.